first slide. ওটার সেকেন্ডটাতে যাও, সেকেন্ড স্লাইড। So the term plasmid, it was introduced by a person known as Lederberg. He was an American molecular biologist, and in 1952 he gave the name plasmid. He also got the Nobel Prize in 1958 in physiology, and with Beadle and Tatum he shared the Nobel Prize. Beadle and Tatum is known for genetic Neurospora crassa genetics. So what is this plasmid? Plasmid, the first thing it means, পরের স্লাইডটাতে যাও, পরের স্লাইডটাতে যেটাতে ব্যাকটেরিয়ার ছবি আছে একটা, হ্যাঁ, what is plasmid? That slide. So basically it is an extra chromosomal circular DNA. The word is extra chromosomal circular DNA. Okay, so in bacteria you don't have chromosomes as such. You have a big, if you look at the picture, first picture, সবুজ রঙের, you have the bacterial chromosome. It's nothing but a circular DNA, and that is loosely termed as the bacterial chromosome. Although you cannot call them exactly as chromosome like we have, like eukaryotes have, bacteria don't have that. Apart from that, there is an extra chromosomal circular DNA present. Not in all bacteria, some bacteria. ছবিটাতে দেখো একটা প্লাজমিডের ছবি, একটা এক্সট্রা ক্রোমোজোম, ক্রোমোজোমটা তো অ্যাটাচ টু দ্য সেল ওয়াল, সেল মেমব্রেন, হ্যাঁ, ওটার বড় আন্ডার মাইক্রোস্কোপ এক্সাক্ট সার্কেল না হলেও basically it is joined at both ends, it's circular. So this is a plasmid basically. Now it is also known as an extra, এখানে সেকেন্ড লাইন যেটা লিখেছি, it contains, it's an extra chromosomal hereditary determinant. So apart from being a DNA, apart from the chromosome, it gives certain characteristics. For example, it can give resistance to antibiotics, because nowadays we see that several of the bacterial disease which we have, they are resistant to antibiotics. That is because they have they incurred this gene from lateral transfer or through mutation or through any other means. They have this gene for plasmid for antibiotic resistance. পরেরটাতে যাও, double stranded DNA, they are circular, they exist independently of host chromosomes. They are not to replicate. Plasmids, in order to replicate, they are not dependent upon the host chromosomes. Host chromosome matlab bacterial chromosome. Okay. So what do they need? They have their own replication origin. Toral line. Replication origin is the DNA sequence which is needed by the plasmid for it to replicate. ওখানে polymerase will bind and it will replicate. DNA polymerase will bind and it will replicate that plasmid like a bacterial plasmid, but the rates may not be the same. So they are self-replicating or autonomously replicating and stably inherited. So if this particular bacteria divides, the plasmids will also divide into two cells. Now next is what is a replicon? Replicon is a DNA molecule that has a replication origin and is capable of being replicated. So a plasmid is a replicon. So plasmid is one single replicon, and the bacterial chromosome, that is another replicon. It can replicate as a unit. Plasmid will replicate as a unit. The bacterial chromosome will replicate as a unit. So they are known as replicons. They have very few genes, less than normally 30, and their genetic information is not essential to the host. So what information these plasmids provide, or what resistance these plasmids provide, say against any antibiotic, it is not needed actually for the host. Only when you put the host in a medium of antibiotic, then the plasmid becomes essential for its survival. But under normal circumstances, it is not needed. But if you give them antibiotic, they will die. So there are the next slide is types of plasmids. First is the fertility plasmids. They carry fertility genes. So bacteria, especially in paramecium, you must have read they have undergone a process known as conjugation, where two different opposite strains they come beside each other. Then a conjugation tube is formed, and from one plasmid, from one paramecium, a particular DNA is transferred to the other paramecium. This paramecium is a eukaryote. Similarly, this thing happens with bacteria also. Okay. If the two opposite strains they come, bacteria side by side, they form a conjugation tube type known as pili, and from through that DNA is exchanged from one bacteria to the other. So there are some plasmids which are fertility plasmids. They contain a gene known as TRA genes, which is necessary for conjugation, and they are useful in transferring genetic information from one bacteria to the other bacteria. Then the most important plasmids which we use are the resistant plasmids. So they are very important socially, and in molecular biology also it is very important. For example, you are making a transgene and you have put it in a plasmid, and this you have put the plasmid inside and you have put them in a media, and you need only the cells which have got the plasmids to survive in an antibiotic media. So only the cells which have got the plasmids, they will survive and grow in ampicillin media or in an antibiotic media. Then you have the cold plasmids. Cold plasmids are plasmids which have this cold region of the DNA, and that is responsible for origin of replication. It's the origin of replication. Okay, you have these type of plasmids. This is just an overview I'm going on the plasmid. Next one, degradative plasmids. Unusual substances are digested. Normally they are not digested. But if you have this degenerate plasmid with this particular genes in the plasmid, then they can digest certain other substances. Now they are trying to find out some enzymes which will which are digested, which digest plastic. Some people have claimed they have found. So these plasmids will be put, and this plastic solution, it is going to be treated with this bacteria. They will break these plasmids, these plastics. They are trying these methods. Then virulence plasmids. These plasmids, when they are when they contain certain genes for virulence, they become a pathogen. Normally, Streptococcus, Staphylococcus is a normal bacteria. It causes mild, harmlessly it lives on your skin. But when they have your plasmids, sometimes you can see acne and all coming out. Acne. Normally it happens in teenagers when they are rich in some hormones, certain hormones. They can be pheromones or other type of hormones. They are rich. It happens in during the teenage years. Yeah. Normally when once you are an adult, you have stable levels. At that time it doesn't have. But that growth, that is very essential for their growth. You can have this, and when you are teen, 12, 13, 14, 15, you will find these bronos and acne coming out. That is because of this particular bacteria. They look like grapes. They have got plasmids inside. Then episome. They are very strange plasmids. So it's a plasmid of bacteria or viral DNA which can integrate into the chromosomal DNA when required. So what these plasmids, so maybe the virus comes, it injects its DNA inside, wants the cell to replicate its viral DNA. But sometimes what happens, these the extra chromosomal circular DNA, they linearize and they integrate inside the bacterial chromosome and become part of the bacterial chromosome. They exist as in a lysogenic phase. Generations after generation, when condition is good, nothing happens. When condition becomes bad, they need to go out. So they come to a lytic phase where they synthesize proteins which are necessary to break open the cell and make new virus particles. Okay, let's go to the next one. So you can also classify plasmids on their ability to transfer to other bacteria. You have the conjugative plasmids, which I have talked about, the conjugative plasmids. They they transfer tragenes. Then there are non-conjugative plasmids also present, and you have partially conjugative, partially non-conjugative plasmid, which are partially transferred, partially not transferred. So they are intermediate classes. So we go to the next slide, which shows a plasmid map, how a plasmid looks like. Every plasmid, in order to survive, it must have certain things. First is an origin of replication. দেখো প্রথমেই আছে origin of replication, and in the plasmid map you will find below the circular plasmid, origin of replication, blue color box. That region of DNA is the origin of the replication, blue color box. Then it must have multiple cloning site. So it must have a multiple cloning site. So you will find that as the restriction site on the right side. Restriction site, দুটো জায়গায়. So you have different restriction enzyme sites. The sequence is such that you can use any enzyme, those enzymes, cut them open, and you can put an insert with the same enzyme and integrate them inside the plasmid. Then you have an antibiotic resistance gene, like I told, extra. Selectable marker it is called. And in red, on the left side of the plasmid, you can see in red antibiotic resistance gene. Normally, ampicillin is used, kanamycin, neomycin. There are lots of antibiotics present where resistance against these antibiotics, protein which this gene makes, gives the cell power resist antibiotics. So it's a selectable marker because say you have transformed a plasmid, particular bacteria with a plasmid. Now, all the bacteria, there are lots of bacteria, all are not transformed. You only want the ones which are transformed. Maybe it has got a gene for insulin inside. You need them, only them. Others agar bada ho gaya, so it's not no good at all. So what you do is you essentially uh, uh, use the restriction enzyme, cut the ends in the multiple cloning site and put them, ligate them, and they are transformed into bacteria. But then to select, you put them in a media which has got an antibiotic. Only those who are transformed with with the antibiotic resistant plasmids, they will survive. Others will die. So they are used as a selectable marker. So basically you need only these three things for plasmids to survive. Origin of replication, multiple polyclonic site, and antibiotic resistance. So the size of plasmids from 1 kb to 4-5 kb, I have even used 8 to 10 kb plasmids for my work. So the bigger they are or very small, if they are, ideal is 3 kb, 2.5 to 3 kb. Perfect, फटाफट clone हो जाता है इसमें. You don't have any problems. The longer it is, chances of cloning, bigger it is, chances of an foreign thing integrating inside is very slim. Stability also has got a problem. And if it's very small, you can put all the sequence together. If the insert size is bigger than what your the plasmid vector, if the insert what you want to enter, then that's a problem. It doesn't normally ligate. So now the next slide. What is a plasmid as a vector? In molecular biology, a vector is different, and in parasitology, a vector is different. What a vector does, essentially in general, a vector is it carries something from one organism into another organism. It's a vehicle, gadi. It's a vehicle to carry something from somewhere to someplace. Okay. So in molecular biology, it is a DNA molecule. It is used to carry genes from organism to organism. So you take one gene from one organism, put it in a plasmid, and you transform it into another organism, make a transgenic organism. But this plasmid is a vehicle, it's a vector. You carry a trait and it's put into another organism. So that other organism organism also tries to express this particular trait which you have put inside. We have seen there are three key parts like origin of replication, multiple cloning site, and selectable marker. These are the three major points for a plasmid. So next slide, go to the next slide. So in a bacterium or in an eukaryotic organism, you have the chromosomal DNA and a plasmid which is extra chromosomal DNA. Imagine the situation. Chromosomal DNA have got multiple genes. Plasmids are known as vectors. So, the vectors can be divided into first expression vectors, shuttle vectors, cloning vectors, and viral vectors. Among the expression vectors, you have insect worm C. elegans expression vectors, you have insect expression vectors, you have bacterial vectors, you have yeast vectors, you have mammalian vectors. Okay, so you can express them in mammals, express them in yeast, express them in insects, express them in C. elegans or worms. Shuttle vectors, you can use them in two types of organisms, in yeast and bacteria, for example. Cloning vectors, you're cloning an insert inside it. It has to have a multiple MCS site, multiple cloning site. And viral vectors. They are viral. When inside a bacteria, it becomes circular. Huh? Shuttle vectors because you can shuttle from one place to the one organism to another. It's like a shuttle. It works here also, it works also here. Among cloning vectors, you have yeast artificial chromosomes in eukaryotes. You have bacteriophages in viral particles. You put the DNA inside virus and let the virus infect the bacteria. That virus will put that DNA inside the bacteria, and you can transform the bacteria by that. And you have bacterial artificial chromosomes. Next slide. So, two most important molecular biology cloning vectors which are in your syllabus are first is PBR32 plasmid and PUC18 plasmid. There are several thousands of plasmids, hundreds and thousands of variety of plasmids. These are the most important two plasmids which you need to study. So next slide, we are talking about PBR322. Now you will see on the left bottom of the plasmid, you see the origin of replication. It has got an ori site. Then the blue color on the right top, where GAMH1 and SAL1 are written, that is the resistance gene, and the resistance gene is tetracycline. It's also tetracycline resistant. On the left side, you have the ampicillin resistant. So it has got ampicillin as well as tetracycline resistance. And you have several restriction sites, PBU1 site, SAL1, BAMH1, EcoR1. So there are unique recognition sequence, and it's got a small size, 4361 base pairs. Okay. This is how the PBR322 plasmid runs. You can practice the diagram. Now look at the PUC cloning vector. Next slide. On the top right, you see a place where it's got NDE1, HG2, NOT1, BGL2, PSU, PVU. You see this site? There is a region known as LAC-Z region. LAC-Z. LAC-Z region. So this has got the bacterial galactosidase gene. Okay, bacterial galactosidase, the beginning part of the gene, the Z part. So what it does is when this gene is present, undivided, complete gene is present, it turns, when you add a substance, a white colorless substance, it turns it into blue. So when the bacteria grows, you can see those colonies coming as blue. But if you insert in this particular region, LAC-Z region, In that lac region, there is a polylinker, and that is the multiple cloning site. If you insert a gene, the gene breaks. When the gene breaks, what happens is that no longer that enzyme is produced, which makes blue colony, the colorless substrate into blue. IPTG, it's known as IPTG, that it doesn't make it blue. So the colonies will be white. So you can see that the white colonies are the ones which have been, which has got that insert which you have put inside. If it's blue, then the whole gene is there. No selection, you don't have the gene of interest put inside. But if it's white, then it's different. That means something is right. Something is right. White is right. That means something is inserted inside. The right gene is inserted inside. And then you on the left side you see ampicillin resistance gene, and it also has got an origin of replication which is not marked over here, but it is beside the at the bottom of the plasmid, that HOE2 site. In this in the HO beside the left right of the একদম তলায় যে HO প্লাস যেটা আছে E2 যে লেখাটা রয়েছে, over there you have the origin of replication. Okay, so these are the two plasmids. So plasmids, as you know, they are used in genetic engineering. You transform them into bacteria. Next slide, next slide যাও. So what they are doing, the ছবিটাতে যেটাতে green part, gene for antibiotic resistance in the plasmid. There is an EcoR1 site. You digest it with EcoR1. Okay, দেখতে পাচ্ছ তো নিচে নিচের দিকে যাচ্ছে EcoR1 digested. So you have EcoR1 sticky ends. The ends are EcoR1 cut sticky. Then on the right of the slide you have the foreign DNA. So the foreign DNA is in between two EcoR1 sites, say for example. Okay, and the pink is the region of interest. You cut it with EcoR1. So the insert is also EcoR1. Vector ends are also EcoR1. You hybridize and ligate it. So you see the recombinant DNA which is formed, which has got both the green antibiotic resistance, the whole plasmid, as well as the pink region, that is the gene of interest. Then you insert or transform them inside bacteria. Along with the bacteria, now this particular bacteria was not ampicillin resistance. Now it develops ampicillin resistance when you put the plasmid with ampicillin resistance inside. And you grow them on the right side, you grow them on a plate with ampicillin or any antibiotic. Only those cells which have got ampicillin resistance will survive. The others will die. These plasmids are used in this particular type of genetic engineering experiments. And plasmids can also be used, for example, human insulin. I am diabetic, I need insulin. Although I am not taking insulin, I take medicines, but doctor eventually will give me insulin. So where is this? In former times when my grandfather, who was also diabetic, they used to take insulin extracted from ox pancreas. So they had to go to the slaughterhouse, cows, sheep, whatever, those pancreas, ox, they used to take these cows, their pancreas, they used to make extract and isolate insulin from there. How much can you get? But nowadays, you take this insulin, human insulin, that was ox insulin, it works the same. But nowadays, you take human insulin, insulin gene, you take it out, put it in a plasmid, put it in a bacteria. Bacteria go over 20 minutes, one bacteria becomes two bacteria. So you put one bacteria, transform bacteria in the morning in ampicillin under resistance, you'll have thousands of billions into 10 to the power 8. Lots of bacteria. So from that maybe you can get as much proteins as you want, as much insulin as you want in the morning. Lots of media, lots of protein. If you want 1 kg, you can get 1 kg. Normally it comes in milligram. You purify that insulin, package it, see how you see how the dose is, or the enzyme activity is, insulin activity is, and package it, standard, that this much insulin will do this much amount of work, you standardize it, sell it in the market. It becomes much cheaper also and more cleaner. So this is one way we can use plasmids. For in the next slide, you see that human insulin production. This, you take the DNA from human pancreas and you insert it in the recombinant bacteria. Then you grow the bacteria, break open the bacteria, isolate the plasmids, isolate the protein, break open the isolate the protein, package and sell. बस अब इंसुलिन दो आराम से पेन के तरह keep on giving insulin. They can be used to make DNA vaccines also. So the DNA has been put in these plasmids, and this DNA is coding for a say for example that particular antibody is a protein. So you get lots of antibody, just inject the antibody and then fine. Done. But there is more. You can use it for there is hybridoma technology used, which I will take later. But you can use it also. These plasmids are only used. So end of class today. Thank you very much.